Hi, this is Brunner, and today we'll be discussing mitosis and meiosis. So beginning with mitosis, it's defined as a cell division that results in two daughter cells, which each have the same number and identical chromosomes as the parent. So preceding mitosis, we have a phase called interphase, which we segregate into three different parts. Beginning with G1, this is where the cell will spend the majority of its life, and where organelles and proteins develop. So in this next phase, we have S, or synthesis phase, which is where DNA replication occurs, and 23 pairs of chromosomes are formed into 46 pairs. Following the S phase is G2, this is a stage preparing the cell for mitosis, and microtubules are beginning to develop and be seen. So after anaphase, we have prophase, and a good way to remember the order of the phases is known as PMAT, which stands for prophase, metaphase, anaphase, and telophase. So beginning with prophase, it's known as a condensing stage, and the microtubules grow even more and become more apparent. Now we have in prometaphase, which is a stage between prophase and metaphase. We have a nuclear envelope starting to disassemble, which is this envelope around the nucleus. And the microtubules even grow larger and begin to migrate to the cells. And at the end of prometaphase, the microtubules begin attaching themselves to the centromeres of the chromosomes and begin to start to tug on them. Now, following prometaphase, we lead into metaphase. And metaphase is where the chromosomes are aligned in the middle, and a good way to remember that is M for meta is M for middle. So, the microtubules are attached, as you can see here, and they're all aligned. Next is anaphase. And anaphase is where the sister chromosomes are pulled apart by those microtubules and they move to opposite sides of the cell. Now following anaphase, we have telophase, which is the final stage. And in this stage, we see the two separate ends of the cell developing, and a process known as cytokinesis begins, and the cell separates and forms two identical cells, as you can see right here, and this will actually even go back to repeat the cycle and go back to interphase. So that's for mitosis. Moving on to meiosis, right here. It's defined as a cell division which reduces chromosomes by half, which are called haploid cells. These four new cells are distinct from the parent cell. And a good thing to note right here is later you will see that the diploid cells have to become haploids, which have half the normal chromosomes. And an example right here can be seen in the human sperm cell and egg cell, which each have 23 pairs, but when combined to form a fertilized egg, get 46 pairs. So the first stage of meiosis is interphase again. And in interphase, we have the same stages, G1, S, and G2. Similar things occur. In G1, this is where it will spend the majority of its life, and then the cell begins to form organelles and proteins. Synthesis, once again, the DNA replicated, but these are now diploids, and we begin to have 46 pairs. And then G2 is preparing for meiosis this time, and microtubules are beginning to be developed. So after interphase, we enter prophase 1, and in meiosis, we separate the phases because they undergo two different types of division. So in prophase 1 is another condensing stage, but a key thing that occurs in meiosis is known as crossing over. And this is what helps develop genetic diversity. And you can see right here in prometaphase, prometaphase 1, microtubules are developed and begin to pull on chromosomes. And we can see these chromosomes like different because they have a piece from each other, and it can be seen down here, right here. So again, the nuclear envelope dissipates. And then in metaphase 1, we have lining up occur again, and all the chromosomes meet in the middle. This time they're diploids. It's a diploid cell. And then anaphase occurs, which is where homologous pairs are pulled apart by microtubules, as seen here. So after anaphase 1, we go into telophase 1, which can be seen right here. And again, you can see this two cells starting to form. We can see the cells starting to separate here, which is known as cytokinesis, and a nuclear envelope beginning to form again. And at the end of telophase 1, cells will have completely divided. Now, going into prophase 2, it is good to know as well, though, that there is an interphase 2, but this is where the cell rests and begins to recover and prepare for further cell division. So now entering prophase 2, we have our two haploid cells right here. And the chromosomes are beginning to condense and microtubules attach to them. Now the chromosomes line up in the middle once again, but are now separated in anaphase 2. And we can see we have unique sides of each cell. Now in the last step of meiosis, we have telophase 2, which is where we can start to see the two cells are starting to each form two sides which are looking unique. Now in the process of cytokinesis, which follows telophase and at the very end, we form 4 uniquely new cells, which are going to look different from the parents because of that stage called crossing over. So that's the end of mitosis and meiosis. Thank you for listening, and I hope you were able to learn. Thank you.